Good evening, everyone. It's nice to see you all. I'm the Adult Services Librarian here. My name is Heath. So I just wanted to say on behalf of the library, hello. This is a friendly reminder to everybody to consider silencing or turning off your cell phones to avoid any sort of disruptions during tonight's presentation. And I just wanted to give a quick shout out. Thank you to Norwood Community Media for being here and making it possible to bring this to a wider audience. Without any further ado, I'm going to go ahead and pass it over.
Start with the vertigo for those on Zoom. I'm just moving you into position. Yeah. All right. Thank you all for joining, both in person and on Zoom. Great to see everyone after the winter on a nice warm day. Choose to come inside and learn about what happened last year in our water quality stuff. So we're going to have two presentations today. The first from Meher Khanna, our environmental fellow, walking through the last year's results. And then we'll hear from Ryan O'Donnell, formerly of the Connecticut River Conservancy, about the display of environmental data on a cool website that he's going to show us. Take it away, Meher.
Okay, hello everyone. How's it going? Good. Okay. I'm Meher Khanna. Nice to meet you. Nice to meet you if I haven't met you before, and nice to see everyone that I already know. Welcome to the State of the Neponset. As Sean mentioned, this is a water quality report. Basically, we're talking about our 2025 data collection and everything, all that data. So I just want to mention we'll have a Q&A after my presentation, but if you have any pressing or clarifying questions, feel free to raise your hands and I can answer those as we're going. Yeah, and I'm sure a lot of you already know about NepRWA and the work that we do, but for those that don't, the Neponset River Watershed Association is a really cool group of people, and we're a nonprofit conservation organization with the goal to advance the well-being of people and nature in the Neponset River Watershed. So we know we do water quality work, that's why we're here today, but we also do greenways work, we do climate change related work, we do advocacy work, go to the State House and tell people to care about the rivers. So we do a lot of different work with the ultimate goal to protect the watershed. And for people that have come to this presentation in the past, you probably know how this kind of works, but something different this year is we're going to make it interactive. So instead of just hearing me talk, you guys are going to talk, and we're going to do that through trivia. Ooh, everyone's so excited. Okay, first trivia question. What is a watershed? So anyone on Zoom or in person, feel free to raise your hand and give us an answer. And whoever goes first. Yeah, what we got?
It's the area of land where all the water comes down into, it's a definition of the area of land that flows into one, in this case, the pond.
Exactly. So the answer was what we just heard, an area of land where all the precipitation kind of drains and goes to the same spot. So this is actually a map of the Neponset River watershed. And you can kind of see all these jagged edges, and the reason for that is because that is the extent of the watershed, and to the other sides of it are different river watersheds essentially. So on this map you can also see kind of a bunch of different dots and stars and everything that represents our different sites, and you're probably going to want to know more, and I'll tell you more. These are the two sampling programs that we have. Our first one is the Community Water Monitoring Network, or SWMN. This is our biggest program with 41 different sites. And these sites are tested from May to October, so 6 months out of the year, on the second Thursday of every month. This is a really big effort with over 50 different volunteers that are helping us out, and a lot of them are here today, so thank you guys. Maybe you might even see your picture over here today. That's because we appreciate you. So yeah, this program is really great. It's been in place since 1995. So pretty great program there. That's our first program. And then we also have a program in sponsorship with Excel Conservation Corps, who's also here today. Here, Excel is a really cool education program that aims to kind of break the cycle of poverty by preparing young adults in the Boston area to get sustainable careers in wastewater operation. So one cool thing that they do as a part of this program is they come to us and they talk to us about, and they sample with us and learn about the work we do. So this group samples at 10 different sites from May to October as well, but on the third Thursday of every month. And throughout the sampling season, we get about 3 cohorts of around 20 students, and this is a newer program we started in 2019. Yeah, okay, cool. So basically, our goals with this program are to, 1, track river health, right? And we're not recording, so perfect. So goals. We want to track the river health because we want to see like historic trends related to water quality, how water quality is changing over time. And in addition, we also take this data and we submit it to the state. We submit it to the Massachusetts Department of Environmental Protection, or MassDEP for short. And they use that data to determine if there's like impairments of different pollutants in the waterways. So our data is really important for that. In addition, NepRWA uses this data just for our own internal decision making. So we know maybe a certain tributary of the Neponset isn't doing as well, so we go do additional hotspot testing there. So that's in terms of river health, but then there's a few other side benefits, if you will. Swimming's a great way for all of you people to get engaged with water quality and caring about the environment. And then also, Excel is a great way for students just to get involved in this field as well and become more well-rounded in their future careers. And here's Dave Bryant, our driver for our samples. We'll get more into that later. But yes, okay, moving on. You thought you were done with the trivia? No. My next trivia question is, we know what we're sampling. What are we sampling for? Any guesses? What are some of the 8 parameters we need to sample our water for? We got one here?
E. coli.
E. coli, we got one. Yes. pH.
Phosphorus.
Phosphorus. Anyone on Zoom?
Dissolved oxygen.
Dissolved oxygen.
Anyone on Zoom wanna?
Temperature.
Nitrogen.
Nitrogen.
Iron.
What was
Say
it?
what now?
Iron.
Iron would be a cool one. Maybe in the future. Okay, we got a bunch of them. Anyone else? Listen, I'll tell you the answer. You got most of them. So basically, the way I have this split up online is like on the slide. I have it split up by where we actually test those different parameters. So some of our samples go to the Massachusetts Water Resource Authority. What they do a lot of different things, but one thing is that they have a lab and they test our samples for us. So they test those samples for E. coli, phosphorus, nitrogen, chlorophyll A, and ammonia. So also if we don't know what some of these words mean, don't worry, we'll get into that soon. And then in-house, so at NepRWA, we test for pH, which is what our lovely volunteer Jim here is doing. And then for our Excel samples, we actually test E. coli at NepRWA as well. Versus for swimming, we test E. coli at NepRWA. And then in situ, which is a little Latin for us, basically means like on site. So in that on site, we test for dissolved oxygen and temperature. Yeah, cool. And I just wanted to give a bit of like an overview about how this program has kind of changed this year. So for our swimming sites, we actually changed 5 of our stream sites in swimming to become pond sites. So you're probably like, why did we do that? Because we wanted to see more, like a more diverse set of sampling sites. We didn't have as many ponds, so we wanted to get more pond sites in our mix. And don't worry about getting rid of that historical data collection because Excel is now sampling those sites. So we just kind of swapped where they were being sampled. And those 5 old swimming stream sites are now Excel sites. So perfect, and Excel's also expanding, which we like to see. And for testing, we are now testing less for ammonia and orthophosphate, and instead doing more chlorophyll A testing. And we're gonna, further along in the presentation, we'll get into why we're doing that, so stay tuned. And I also want to mention that we have 4 Sentinel sites. And these sites, Sentinel basically means that we're testing these 4 sites for everything. So all those 8 parameters you mentioned before, that's what those sites are being tested for. And we have 1 site in each section of the river. So kind of if like we see a problem in one section of the river, we kind of can isolate where that problem is happening. And then duplicates, just for some context, we sample all these sites, but just for proper scientific procedure and everything, we have some duplicate samples, which are basically just the same sample but taken twice. And I just want to mention this because a few volunteers reached out to me and were like, why am I getting so many duplicates? And oh, sorry. The reason that you're getting more duplicates is because maybe you have a Sentinel site. So now the duplicates were always based on like what tests are happening to the sample you're actually taking. But if you're now we kind of change what sample is being tested for what. So like some sites will get duplicates more often than others. It's kind of a yeah. Sorry, my laugh's too loud. Okay, moving along to our first parameter, E. coli. Okay, another trivia question for you. Why do we test for E. coli? Anyone? Yeah?
Fecal contamination.
Fecal contamination, yep. Yeah, essentially, yeah. Good, you guys, this is easy for you. Yes, so it's an indicator for fecal contamination, and that means pathogens, which is not good. So E. coli, for some context, is the coliform bacteria found in the gut of warm-blooded animals, like all of you, and like our pets, our dogs, and stuff. So I just like to mention this because it's an indicator, but it's not inherently dangerous. Okay. And some sources of E. coli are like animal waste, but also various like, human-related wastewater, and then generally proximity to impervious surfaces. And now, talking about the E. coli standards, this whole topic gets a bit complicated because there's a few different standards that we think about in terms of E. coli. So the most up-to-date standards, according to the 2024 state standards and the 2012 EPA, the Environmental Protection Agency, the federal standards, are a bit complicated. So these standards are more geared towards frequent sampling, and they use a geometric mean and a statistical threshold value, which kind of limits, it kind of just creates like this site fails or this site passes. And we want to see more of that nuance. So what NepRWA does is we like to use a single sample maximum, which basically says, okay, this site is, if it's above this level, then it fails or it passes. And then I'll show you in the next slide how that is a little more nuanced. But I also want to highlight that the state is actually currently changing their standards and they're updating them to make them stricter. So you'll see what I'm talking about soon. So these standards are referring to our single sample maximum. So basically, we're saying that these standards are based off of human health for recreation. So if we're above 235, then we can't swim in it, if we're above 235, we can't swim in it, and if it's above 1260, we can't swim in it. And I also want to give this little disclaimer that, NepRWA, we're testing water quality, we're testing historical trends, as we know, but sometimes if you want the most up-to-date information, it's always good to check with your town as well. And you're probably like, what is this giant unit that means something to me? Well, I'm going to ask you the same question. Any guesses? We got a guess, yeah?
Colony forming units?
Colony forming units, and the last part.
Milliliters?
Yeah, exactly. So nice. This is a hard one. So colony forming units, basically, of the bacteria, per 100 milliliters of water sample. So this is going to be a concentration of E. coli in the water, and so that's kind of helping us determine the level of E. coli. Makes sense. So generally for E. coli, we say that the main stem does pretty well, but certain tributaries are consistently not doing as well. And a quick plug, if you want to learn more about this, come to our annual report card event, which is in partnership with the Mystic and the Charles River Watershed Associations. It's happening in May. So if you want to learn more about that, talk to us after as well. Okay, now let's finally get to the data, the reason we're here. So here is our data. So take a look, but let me get you situated with what's happening here. Basically, each column represents 1 month of sampling. So, and the different colors represent meeting the different standards. So the green represents meeting the swimming standard, the yellow represents meeting the boating standard, and then black represents failing, meeting neither standards. And generally, what we're seeing, the trend is, wow, July is pretty bad. And the reason that is the case is, I don't know if you guys can see at the bottom here, it's a bit small because I didn't want to take away from the graph, but you can see that is the precipitation for each of our sampling events. So July, the swim and samplers out here know how bad July was. That was the day it poured. July was pretty bad, and the data proves it. So the exact same thing is happening here, where we're seeing a lot of more failures due to that precipitation, because more runoff means more of that, like the stuff on dog poop and getting into the waterways, more potential wastewater contamination. So all these things are kind of leading to those failures. And even in October, we see a bit more of a dip in failures because there's a little bit more water as well. But yeah, makes sense. July was crazy. And that was looking at our single sample maximum. This is looking at that standard that I said was kind of weird, which is the geometric mean. So basically in this, every single column now represents a site. So feel free to look for your own site up here. And the colors mean the same thing, right? So the black means it fails, yellow means it passes boating standard, and then the green means it passes our swimming standard. And this, for people in person, this is the graph on your sheet, on your seat. The sheet on your seat, yes. Question?
This is for the whole season?
Yes, for the whole season. Well, I've got a question, yes.
Sorry. It says for meeting voting standard less than 630 CFU, but on this other slide it was 1200.
Oh yeah, so this is looking at our standard for geometric mean, which is different than that single sample maximum. Yeah, good question. Yes, so I'm gonna go back a slide to show you. This is looking at our single sample maximum, which is the standards I mentioned before, and this is kind of the weird standard, the geometric mean. Yeah. So another follow-up question.
I'm wondering if you're seeing any changes as towns start to pursue the illicit connection work that they're required to do under the MS4?
Can you repeat the question?
Yes. The question was, are we seeing changes with towns doing illicit connection work and trying to reduce that work, or trying to reduce the illicit connections and
Trying to address it.
yes. Yeah, definitely. And you can also look at my poster later, which is about Meadow Brook, the lowest scoring tributary in the Neponset for E. coli. And Meadow Brook has a history of leaking under drains, or leaking sewer pipes going into under drains and going directly into the brook. And we do see some progress, but these things take a long time. And this is historic quality monitoring. Okay. Yeah.
Can I ask a question?
Yeah, if it's a clarifying question, yeah.
Oh, no.
Okay. sorry.
We'll
Yeah.
have Q&A afterwards.
Yes,
Okay.
Q&A afterwards. Plenty of time. Okay. Yes. So I want to show you guys. These are the current standards, right? The current geometric standards. But I mentioned earlier, the state is actually in the process of updating these standards. So Get ready for it. Whoa, these are the new standards. Yikes, yikes is right. So these are the new standards. Yeah, it's a lot stricter. You can see this bracket represents kind of all the sites that would now be failing under the new standards. So it's like stricter. Yay, government works sometimes. Okay. Yeah, okay, cool. Moving right along, we're gonna go into the next parameter, which is phosphorus. Phosphorus is an essential element for life, and it's often the limiting nutrient in our freshwater systems. And phosphorus, when there's a lot of phosphorus in waterways, we see this process called eutrophication happening. Eutrophication, basically, algae starts to grow like crazy when there's phosphorus in the water in our freshwater systems. And when this algae starts blooming like crazy, then it keeps going until there's no more phosphorus left. And then when the phosphorus runs out, the algae starts to die off. And then that process, when the algae starts to get decomposed, then it uses a lot of the oxygen in the water. So we see a lot of dissolved oxygen going down. We see fish starting to die off because there's no oxygen in the water, leading to lower biodiversity, a lot of things. And harmful algal blooms are basically like when the algae that grows is it has like toxins that are being released, which can also happen sometimes and can be unsafe for those organisms and humans and everyone. Okay, yes, so that's phosphorus. And some sources of phosphorus include like fertilizer, include wastewater again, kind of similar to, I was mentioning E. coli, and also happens naturally, but yeah. And for our standards for phosphorus, this is based on the type of water body. So for our flowing water, we see kind of a higher threshold for the phosphorus. And then for our still water, we see a lower threshold. And then I just want to mention there's also this kind of other threshold down here, which is for rivers entering a lake or reservoir. And it's kind of in between, but NepRWA has unclear guidance from the state about what this means. So we kind of don't use it, but we use the other standards. So you might be asking yourself, why are there different standards for the different types of water bodies? Anyone have a guess? Anyone online?
For example, at the seasonal, what time of the day that you see that the Escherichia is more? What time of the day?
For E. coli?
Mm-hmm.
So the question was, what time of day do we see the more E. coli?
Yes. Exactly.
Yeah, so generally what we see sometimes what we see when there's like wastewater contamination in a stream is like in the morning and like in the evening, kind of when people are at their houses using water, we see kind of increases in E. coli during those times. As a general trend. We are testing, all of our samples get tested at the same time every day, so we don't really have that kind of data, but does that make sense? Answer your question?
Okay, is there a difference for the season? For example, in the case of the monitoring, we have dry season, wet season. Okay, in what season for this river, we observe the increase of the ratio?
Yeah, so one of the main reasons why we test May to October is because that is that is like the main time people are out in the water, and also that's when the bacteria will like be alive in that water. If the water gets too cold, the bacteria won't survive in the water. So kind of during the summer months, we see more bacteria than during the colder months. Okay. So let's go back to this question. Why does the standard for phosphorus vary for still versus flowing water? Got to answer in the back.
I'm not sure, but is it because it has more effective as more water?
So your answer was because it, can you say that again?
There's more of it.
Right, okay, so the answer was there's more of it, and basically. So stagnant water is more sensitive to algal blooms and plant overgrowth. So that phosphorus stays in the water for longer than it would in like a still waterway. So that means more algae can kind of bloom. So yeah, you got it. Okay, cool. So that's phosphorus. And generally for phosphorus, like we just kind of mentioned, our rivers pass, our lakes and ponds fail because of that reason. And let's see what our data shows. So, this is basically the same kind of layout as our E. coli, right? So these are just for our flowing sites. So we're seeing generally it's passing, but July's not doing too well. And you already know the reason. The reason is precipitation. The same kind of same ideas that we mentioned before. So we're seeing more failures due to that increase in precipitation for phosphorus. So this is our flowing sites, but I want to show you what our pond sites look like, which is a lot more failures. And we're not surprised by this. This is the trend because what we just mentioned, right? There's going to be more phosphorus in those still waterways, and yeah. Okay.
What happens with September there? Unclear.
Yeah, I think that's kind of unclear. Probably more just within the range of uncertainty and chance. Yeah. Don't have a good answer there. Yeah, so that's our phosphorus. And moving right along to chlorophyll A. This is a fun parameter because we're testing more of it this year, like I mentioned earlier. So for some context, chlorophyll A is a pigment used in oxygenic photosynthesis. And it's found in algae, so we actually use this as a way to measure the biomass of algae. So this depends on a lot of different things. It depends on nutrients like phosphorus, how much phosphorus is in the water, and other things as well. But especially, it's linked to phosphorus in our freshwater systems. And the reason why we're sampling more for chlorophyll is because we want to get a nutrient impairment for the Neponset River watershed. And what Massachusetts Department of Environmental Protection wants to see is not only is there like an increase in, or not only is there a high level of phosphorus in the water, but there should also be a high level of chlorophyll A. Because what the state wants to see is both of these happening in parallel, meaning that one is kind of connected to the other. So they want to see a result of an excess of phosphorus in the water, leading to kind of plant growth. So that's why we're testing kind of more for that. And so for our standards for chlorophyll A, we want to see our waterways being less than or equal to 16 micrograms per liter. And here is a graph for you. So this is a different graph than we had before. This is a box plot. So in each box here is representing a different site. The first box you can see is a lot. It's taking up the entire graph here. So what this kind of tells us is Croc Pond has a lot of algae, and here's the proof. Yes, and actually this pond already has a phosphorus impairment, just more so due to like historic industrial kind of happenings in the area, not due to like non-point source pollution and stuff. But yeah, so we already know Croc Pond is a problem, so let's get rid of that. And just look at every other site besides Croc Pond, and we can actually see some of the data. That's nice. So it's the same idea here, that like I have 16 micrograms per liter line here, which is our chlorophyll standard. And then the way I like to read a box plot is I look at that black line in the middle, this black line right here. That represents the median of the of the chlorophyll A of that sample at that site. So you can see that Man's Pond here, that first site over here, is basically more than 50% of the time Man's Pond is above the chlorophyll A standard. And these other sites, all these other ones that have like these dots or these lines that are above the line, that means that occasionally they have been above the standard, or at least one sample, at least some samples throughout the season have been above that standard. I got a question. Yes.
So is you're averaging across all 6 sample times over the summer to get that average?
No. Sorry. This box plot is showing kind of the range of the samples. So like this is right here, this line at where this line kind of starts is the lowest sample, the lowest amount of chlorophyll that this site has.
What are the
Ever
multiple
had?
samples though? Multiple samples in 1 month or multiple months?
Is that all in 1 year? Is
Yeah.
that 2025? Is that,
This is all 2025.
okay,
Yes.
so it's all 6 samples for 2025 inside that box?
Yes, exactly.
Can you just repeat that for the people?
Yes, so these are all 6 samples from 2025 represented in this box. Yeah. Does that make sense? Sorry, I don't know if I explained that right. If I explained that well.
Yeah, so
Another
each-
question.
Each box has 6 samples in it.
Yeah.
And the median represents, the
3 above, 3 below.
Yeah.
Yeah.
The line in the middle is the median.
The line in the middle is the median. Yeah. Sorry, hope that makes sense. Yes.
And then the far-reaching dots, those just outliers, you're not sure? Like,
Outliers, yeah, but you could imagine that like this line is like being drawn here because it's basically the same idea, but they're kind of pretty far away from the, yeah, don't want to draw really long lines. Okay, does that make sense? Okay, and other thing I want to bring up here is you can see, so all of these sites are our pond sites, right? Up until here, and then all of these sites down here are our flowing sites. So you can see like all of our pond sites have a lot greater level of chlorophyll versus our flowing sites. Yeah, and okay, that's what we see from this graph. So that's for individual sites for chlorophyll. Now, I mentioned earlier that phosphorus and chlorophyll have a relationship that we want to look at. So here is actually a plot looking at total phosphorus on our x-axis here at the bottom, and then chlorophyll A up here on the side, on the y-axis. And all of these different dots up here are our different sites being tested for, or all of our different sites. And we have our flowing sites in orange and our blue sites, our pond sites in blue. And you can see it's kind of hard to see all this data because it's very clustered down here and it's kind of far spread out. So what we did is we decided to log transform this data. And we just did this so that we can kind of see the full extent of the data a bit better and kind of look at some of the trends a bit better. So this is still showing us basically like flowing sites and pond sites, and this line here is again our chlorophyll standard. And then we have 2 vertical lines over here that represent our different total phosphorus standards, right? So this is our total phosphorus standard for our flowing sites, and then this is our total phosphorus standard for our pond sites. And we already mentioned earlier that our flowing sites, basically, we aren't as concerned about them because you can see all of the orange dots are like way down here. They never exceed our chlorophyll standard. So that's not really an area of concern. So what we did is we decided let's just look at the pond sites because that's where we see the biggest impact. And we actually threw up a regression line on there, or like a trend line to see how that relationship between total phosphorus and chlorophyll. And we can see like, it's generally a positive trend. With more total phosphorus, we see more chlorophyll A as expected. And again, we have our chlorophyll A standard over here, and this is our total phosphorus standard. So I just wanted to mention that basically everything kind of here, so everything in this kind of box here, is basically exceeding both of those standards. So this is kind of what the is really interesting, and it's kind of what's going to be of most concern to the state and to us.
That's a question.
Yeah.
So that line is the result of all the analysis during all the seasons? Wet season, dry season, is the result?
Yeah, so this is taken, so the question was, what are these data points coming from, like seasonally and depending on if it's rainy or not. So this is taken May to October.
Okay.
And we had a few, based on, if you remember the other graph I showed earlier with the E. coli, July and October were kind of our biggest rainy events. So we had, we have some rain events in there, and we have some dry events as well. Yeah.
Is there a correlation between the wet season and the total phosphorus?
So the question was, is there a relationship between total phosphorus and like wet weather? And yes, we see
Wet
with
season. Wet season.
well the I mean, what's the wet season considered?
No, it depends on the time of the year.
We're in a drought. I think it more so depends on. I'm not exactly sure. I think it more so depends on like if it's raining, and because I feel like we don't have a general wet season here in the state, right, Sean?
Yeah. We tend not to have wet and dry seasons. We have more warm and cold seasons. So when it is wetter, there's more runoff that carries the phosphorus.
No, when we have a lot of runoff.
Yeah.
Okay.
So when it's raining, so we can also look at this by rain, but we can look at it a million ways.
Yes. The answer was we have more warm and cold seasons versus like wet and dry seasons, but we do see with wet weather we see more phosphorus in our waters, which would also lead to more chlorophyll.
Yeah.
Kind of, yeah.
And the chlorophyll problem is worse in the warm weather when the algae can grow well.
Right, and yeah, exactly. And the chlorophyll is worse in warm weather.
You have a question on Zoom.
Okay.
Can you go back to your first chlorophyll slide?
Yes. This one?
Yeah. Maybe the second one.
Maybe the second one.
So Dave Bryan said that the takeaway for the first slide for chlorophyll seems to come to what the graph actually showed. I see. Because your takeaway there is that ponds are generally exceeding the standard. I think.
Oh.
Do you want to address that?
Yeah. So what I meant, I guess, more so was the ponds have higher chlorophyll levels in general versus our flowing sites, which you can see how low the flowing sites are here. But our ponds, like this is exceeding the standard, and then these ones occasionally, like we see some samples that are exceeding the standard.
Yeah, and I was just going to add to that.
Yeah.
For Dave online. In the room, obviously, we're drawn to the median, but technically by the state standards, none of these should exceed the standard ever. So 4 of these 6, 7 sites exceed the standards at least once during the season, which should not happen.
Right. Yes, that's a good point. So.
Okay. Thanks for that clarification.
Yeah.
Okay. Cool. Gonna move along. We saw these graphs. We were at this graph. Positive trend, and these are concerning. Okay. Gonna keep going to our last parameter, which is dissolved oxygen. So dissolved oxygen is the oxygen present in water that organisms can actually use, and it tells us a lot about, like, can this water body support the organisms that are living in them. And this is what we use to actually sample the dissolved oxygen, and we actually have the probe up here at the front for people in person to look at as well. And some sources of dissolved oxygen are diffusion from the atmosphere, so naturally some oxygen gets into the atmosphere, and then through aeration from like rocks and riffles like you see in this picture, and also naturally through photosynthetic activity, we see dissolved oxygen getting in our waterways. You thought you were done with the trivia questions? No. So, next one. What type of water do we think holds the most dissolved oxygen? Cold or warm? Flowing or still? And why do we think so? Any guesses?
Cold and flowing.
Yeah, got an answer here?
It is cold and flowing.
Cold and flowing? You're right. Good job. So, cold and flowing water holds the most dissolved oxygen, and that is because if it's flowing water, like we saw in that last picture, there's more aeration and more oxygen can actually get into the water. And then cold water means that if you think about like what cold means, it's kind of like the molecules are moving around less than they would if the water was warmer. And that means that oxygen's like more likely to stay dissolved and less likely to kind of go out of that water. Okay, and so for our dissolved oxygen standards, we kind of assess dissolved oxygen based on, like we mentioned kind of earlier, related to the fish that can be supported by the waterways. So we have 2 types of fisheries that have different standards. We have a cold water fishery, which has which we want around 20 degrees Celsius, and then we have a warm water fishery, which is the 28.3 degrees Celsius. And we can see that our warm water fisheries are not as sensitive. So that's why they actually have a kind of lower threshold for how much oxygen is necessary for those fisheries. And if you're wondering what is a cold water fishery in the Neponset that we actually test for, here's the answer. So these are basically all the ones up here are cold water fisheries. Everything else is going to be a warm water fishery. And here's the data. So on the left is our cold water fisheries, and on the right are our warm water fisheries. And we're kind of looking at if it's meeting or failing the individual standards for each of those. And generally, from this, we can see it's doing pretty well overall. But we do see with warmer months, due to summer months, when it gets warmer, we see less dissolved oxygen in those waterways. Oh, question? Yes.
On the cold water fisheries, is that based on the sampler's temperature readings, or is it based on something else?
Yeah. So we have a dissolved oxygen probe, and samplers put that directly into the water and sample on site the dissolved oxygen, and that's what these energies come from.
But the cold water fishery is based on temperature?
So the cold water fishery is like a definition by the state. So if we go back, these would be defined as the cold water fisheries.
And one of those
yeah.
is my site, and it's right past a very shallow pond which heats up the water quite a bit. So I'm wondering why it made it into the cold water versus the warm water, because it doesn't seem to be terribly cold compared to the stuff upstream.
That is a good point. And so though this is kind of like the definition of what a cold water fishery is, obviously with climate change, with all these different things, we're seeing temperatures kind of exceed what should be for a cold water fishery, and that is an issue. And but it's still defined by the state as the standard. So that's kind of it's still defined by the state as that type of fishery. So we still follow that standard with it. But you're right, it is a problem. And yeah, Sean, you want to add something?
Well, Ryan on Zoom has added that they're usually defined by geography and elevation. But also I would add that technically speaking, the Neponset doesn't have any cold water fisheries by temperature alone. But instead what they have are what Mass Wildlife calls cold water fishery resources, which are streams that have cold water fish. So none of our streams have enough, either have the proper temperature or have enough data about their temperature to make them cold water streams. But they do have cold water fish like brook trout.
Yes. And
under state law, they should be protected like cold water.
Yes. So yeah, well, even though they're not technically defined as cold water fisheries by the state, they should be still held to the same standard, even though with climate change and things, we are seeing them not meeting these standards, which is unfortunate. Yeah, question.
How many of the sites do you take a dissolved oxygen measurement each month?
All of our sites. Right. Oh, yes, yeah. Sorry. The question was, how many of our sites do we test for dissolved oxygen at? And we test dissolved oxygen at all of our sites.
Oh, because I've never been given a meter on it. I
right.
assumed it was like a subset.
Yes, so that's a good point. So for our samplers out there, maybe you take an individual water sample, but then someone else comes and they with their dissolved oxygen probe, and they test for dissolved oxygen maybe after you, or before you were even there.
One morning I did bump into someone.
Yes. Yeah.
River.
Yeah, and our dissolved oxygen monitors, they usually go to multiple different sites. Because it's you stick the probe in, and it's generally it pops up the number pretty quickly, and then they kind of go to a few different sites and kind of do like a route with our dissolved oxygen testing. So
on sampling morning, there's somebody else who's doing all the sites, or a subset of the sites.
Yes.
Each person, but they're all covered that morning in the water, not from our samples later that day.
Sorry, I didn't get the last half of your question. I'm not repeating the question, so let me do that.
Oh, yeah, so there is a individual who hits all the sites, maybe multiple sites per person, taking measurements in the water and it's not being measured later with our samples in the bottle, correct?
Yes, so the question was, how does the dissolved oxygen kind of work in parallel with our regular water sampling program? No, it's okay. Sorry, okay. So basically we have our water samplers that go out and take our individual water samples that usually take one sample at one site. And then we have a few different dissolved oxygen monitors that go around the same time, and they go to like around 5 or 6 or 7 different sites, and they sample the dissolved oxygen kind of around the same time.
Yeah. And then the Excel team, they do their own something like that.
Excel team, they do it all at once. So yes. Okay. Hope that made Hope that made sense. If anyone online is confused because I didn't repeat the question, let us know. Okay. So that's the data. And we're basically at the end. So I want to give a little bit of an overview. We like doing historical water quality trend tracking because it's important to submit this data to the state to advocate for our river and tell the state that different parts of our river is impaired for different pollutants. And so that's kind of the point of doing all of this. And then what we generally see with our E. coli and phosphorus is July was the worst of it, and that was due to an increase in precipitation, leading to more runoff, leading to more runoff from like dog waste getting into the water, more potential wastewater contamination due to all of this precipitation. And then with chlorophyll, what we see is we are sampling more chlorophyll this time around, or this year, because we want to get a nutrient impairment for the Neponset River watershed. So we're sampling more for chlorophyll A, and we want to see that correlation between phosphorus and chlorophyll A in our sampling. So another little trend that we're seeing with phosphorus and chlorophyll A is that our ponds are doing worse and exceeding the standard. And then finally with dissolved oxygen, we see mostly passing, but with seasonal variations, warmer summer months, we see lower dissolved oxygen. Okay, question, yes.
So why do you want to show nutrient impairment with your chlorophyll A?
Yes, that's a good. So the question was, why do we want to see a nutrient impairment with the chlorophyll A? And that's because if we have a nutrient impairment in place, then towns and cities are kind of required to do a certain amount of mitigation of that. So they're required to put in more stormwater stormwater infiltration and stormwater pollution prevention stuff, and and required to do more outreach work as well. So, do you want to add to that? No.
I was just gonna say.
It can prompt a remedy is what it is the short answer, I would think.
Right.
Yes.
So in theory, we don't want a nutrient impairment.
Right.
If there is one, we want it recognized.
You want it recognized, yeah, exactly.
Yeah.
You want
Another
to show
question.
it.
Yeah, I hear you talking about stormwater. What about toxins from old industries and stuff like that in the Neponset River?
Yeah, so the question was, I mentioned stormwater, what about toxins from old industries in the watershed? And like I mentioned with Crack Rock Pond, actually, that already has a nutrient, has a phosphorus impairment. And so we do see kind of with some like historical kind of pollution and historical industrial kind of waste getting in our waterways, we do see some impairments already existing without kind of our data, because we know of those problems. But yeah, this data is kind of more looking at non-point source pollution. So kind of all those things I mentioned I mentioned the sources of these kind of pollutants. That is more what we're targeting with this data collection. So, yeah.
Isn't the EPA all over that by now?
For Superfund, EPA is all over that. Yes, question?
In the case of water quality, it's very important to mention the value of the pH. Why in this takeaway we not mention it?
So the question was, why was the question like we test for pH? We test for pH, but I didn't really mention that today. Is that why you brought that
No,
up?
I'm talking about the takeaway. all the step that we have to, that's some easy indicators
Mhm.
that we have here.
Mhm.
But the pH is very important for, as an indicator of the pollution.
Yeah, definitely. So the question was like pH is an indicator of pollution. It's very important. So we should maybe talk about that more. And generally, the pH is generally within a normal level. If there's kind of a lot of a certain contamination happening in that area, like if there's some sort of like a, I don't know, I can't think of anything right now, but like a spill of something, like yeah, kind of like an event that happened where the river's contaminated, then we might see more of an the pH increasing. But generally, from kind of the pollutants I mentioned in this presentation, we don't see too much variation with pH. It's generally still within normal levels. But for example, like in Meadowbrook, there was a construction happening a few years ago, and the entire brook turned white because of all like this construction material getting into the waterways. So something like that could spike the pH, but generally with normal kind of pollutants, we don't see the pH varying too much. What do you think, Sean?
Yeah, and I would say it's a really important thing that we monitor. For most of our sites, most of the time, it's not a problem. And so just for sake of time today, we focus on the things that do tend to change.
The presence of the E. coli with the comparison of the pH can help us to understand the source of the pollution.
Yeah, and we're interested in doing that. The problem is that we're trying to get a 30-minute presentation of all of our data, so we just kind of condense some stuff down. But it's a really good point.
I'm already like 20 minutes over. So, okay.
Speaking of, we'll just, I think we've had conversation as we're going, so we'll let you wrap up and then we'll switch over to Ryan.
Okay, yes. So, gonna do say a quick thank you. 30 years of swimming done. Yay. Thank you to all our swimming volunteers. Yeah, you guys are amazing. We wouldn't be able to do this work without you guys. And a few quick plugs. Our cleanup is coming up on April 25th. Everyone should come. Ask me if you have questions. And QR code's up there. And
Can you order better weather this year?
I'll try. Yes, so cleanup. And then another plug for something happening like next week is on March 16th, Storm Marty Stoughton. If you want to learn more about that, let me know. We can talk about it. And a quick thank you to everyone here. Thank you to the swimming samplers, swimming DO monitors, swimming drop-off coordinators, swimming site coordinators, the NepRWA Deer Island Lab. Thank you to Excel. Thank you to Lisa. Thank you to Don. Our Excel student volunteers, the Norwood Library here today, the cable company that I forget the name of. Thank you to the NepRWA community. Thank you to everyone here. Thank you to NepRWA, and thank you to my boss, Sean McCanty here for helping me with R, helping me make these graphs. And any questions? Well, actually, we already answered a bunch of questions, so we're going to move on because we don't have time. Okay, and because we're running low on time, we're going to jump to Ryan's presentation. Ryan, feel free to share your screen, and I will stop sharing mine. Okay.
It's a little bit quiet. Right now.
Can you hear me?
Yes.
Yes.
Is that better? All right. You'd think the headset might work better, but okay. Thank you all for having me. I wish I could join you in person, but I am thankful that I didn't have to drive the 2.5 hours from my home in Heath, Massachusetts, where we still have several feet of snow. And a lot of mud. Okay. There we go. All right. So I'm calling my Zoom controls are in front of my notes. I call many of my presentations, but can I swim in it? Because that's a question I get asked the most. And normally I'm presenting many of the things that you just heard about. What are the different water quality parameters? What do they mean? And do they affect whether or not you can swim or boat or fish or whatever? But today I'm actually going to talk about communicating. So first I'm going to introduce you to me. I'm going to go over some of the communication challenges that we face with water quality data, and then a brief case study and a bit of a brag about how I designed one of my favorite charts of all time, and then also going to go over revamping the Connecticut River Conservancy's Is It Clean website. So who am I? That's me sampling the Deerfield River for chloride last winter. It was quite chilly. But I earned a BS in geology and a master's in sustainability sciences with a focus on water policy and climate change from UMass Amherst. And that's where I first started getting into rivers. And immediately before I did my master's, I guess I knew I was into water at least a little bit because I volunteered in Connecticut River Conservancy's water quality lab testing E. coli samples in 2012. And then as part of my master's, I interned with the Vermont Agency of Natural Resources, and it was through that I was first exposed to the sampling side and supported the Southeastern Vermont Watershed Alliance, aka SEVLA, and the Ottaquechee River Group with their volunteer monitoring programs. And then since 2014, I continued coordinating the SEVLA programs and working with CRC. In 2017, I actually started a brand new monitoring program on the Deerfield River, which is my home watershed. And that was a great initiative because it was the first that really covered both Massachusetts and Vermont. The watershed is about 50-50. And then from 2018 to 2025, I was able to bring that all together as the Connecticut River Conservancy's Water Quality Program Manager. And so I brought those programs with me that I was already running and took over the Massachusetts and Connecticut monitoring programs for CRC. First, water quality communication challenges. I was looking for a picture that communicated too much data, and so you Star Trek nerds might recognize data. And it was too funny of a picture to not use, so hope you'll forgive my silliness. But that's really the crux of it, is that there's always a lot of data. So why is it hard to communicate about water quality? We want to, like, as the scientists doing the work, we want to communicate accurately and precisely. We pick very specific words on purpose because we don't want to be misinterpreted. And in my time at CRC, I ran into this issue a lot because I would send a very carefully crafted paragraph about water quality to our communications department, and then they would be like, this is a little confusing, and they would simplify it, but then it meant something completely different. And so it's a really difficult balance to avoid jargon while maintaining that precise definition of things. Also, audiences have varying skill levels of interpreting data. So we're trying to reach the people who know almost nothing about statistics and environmental science, and we're liable to have complete, total experts, people who know more than we do about what we're trying to talk about. And so some people want simple information. That question, okay, but can I swim in it? And others want to see every single number and make the conclusions themselves. And so it's kind of hard, because if you throw a bunch of numbers at people who don't know how to interpret it, they might be turned away or just confused or make the wrong conclusion. So that is another area that's difficult to balance. And actually, we saw this in the last one, and I appreciated that this happened, which is often we need to use a logarithmic scale, but people really have a hard time understanding that. And so it does help. This is actually something that I do all the time, is I show it with a linear scale and be like, you can't see the data, so we do this thing, this is how we're looking at it, but also it's just so much, everything we do normally is more linear than logarithmic. Water quality data is complex and it's noisy. There's so many external factors. We talked about a lot of them today. Land use, weather, flow, time of year, temperature, if you have a combined sewer overflow. There's just so many things that can be happening. And so there's just all this noise in the data. And so it's kind of hard to understand sometimes, is that outlier an outlier? Or is it something that happens once a week because the specific thing happens in the watershed? Audiences often want quick, decisive action. So, we would occasionally have a really pristine swimming hole, in the mountains, and normally the E. coli is really good, but suddenly you have a high spike. And is it that something changed, or did a goose fly by and poop in the water right before you took a sample? Well, you have to wait till you take your next sample till you find out. But sometimes seeing that high number is really concerning, and people are like, we need to do something now, we need to solve this problem now. But is it a problem, or was it a stray goose? I think another challenge is that data can be both overabundant and sparse. So some types of monitoring, like continuous monitoring, can produce thousands, tens of thousands, hundreds of thousands of data points. You can be monitoring at a lot of different sites, but maybe infrequently. You miss samples because of real life getting in the way. So there's just a lot of gaps, but then also sometimes there's too much data to look at. You don't have time to go through the pH. And then also another thing is, us water quality professionals are looking at this stuff all the time. We're so used to it. And so what seems simple and common might actually kind of be a niche bit of knowledge. So I have this example here. This chart, I bet some of you look at this chart and you go, I know exactly what that is. And some of you are going, I have no idea what it is. And it's in Celsius. I'm still bad at converting Celsius to Fahrenheit, but this is actually just a random, just how temperature logger data comes to us. And so, as you see, that's a lot of data. It comes to us looking like that, and then it's up to us to interpret it. Also glad to have seen this happen, which is, I always start from the basics. I start with this image. I have no idea where it came from. It just lives on my computer permanently for the past 14 years, but it's my favorite diagram of the watershed because I think it communicates a lot in it, but also it's interesting enough that if you already know what a watershed is, you're not too bored because you're looking at, Sasquatch or bear, or wondering why the tuna are so big in this harbor. which is to say that one of my approaches to communicate about this is assuming that at least one person in the audience is starting from scratch, and it never hurts to get a brush up on your knowledge. All right, so first I wanted to talk about creating clearer charts. I think I love making charts, and it's really easy to make a very confusing chart. So, what's the problem? Well, often the problem is food. I'm sort of known for using this emoji in my presentation, so I couldn't leave them out. But the problem I was trying to do is address is how do I present 5 years of E. coli data clearly. We have samples that are collected by volunteers over 5 years in 3 different states. Connecticut and Massachusetts were sampled on different days of the week. Vermont was sampled on the same day as Connecticut, but only every other day, or every other week. And so often I was used to showing data by the sample date. And if all your sample dates are different, how do you do that? And then also that weather conditions might be different across such a large area. So I'm also used to presenting data as either wet weather or dry weather. And if we're looking at such a large geographic area, even on the same day, you might have different weather conditions. And I wanted something that I could include in a report for colleagues, volunteers, and the public to review on their own, so I wouldn't necessarily be there to explain it. So if I just throw 1 year of data into a line chart, I could put it on a bar chart. It's overwhelming. We have way too many sites. What's a good result? What's a bad result? Because it's a linear Y scale, which means each of these bars is equal to the same amount. The good results and the bad results are actually right next to each other. Where are the sites? You can probably figure out what state they're in. And I actually think that CRC's site IDs are fairly useful. They tell you the state, which river it is at the river mile. But for most people, it just looks like gibberish. So here's a bigger look. I also have the wet and dry weather. But so this is not like a helpful way to look at the data. And then if you try to do it for all 5 years, adding more to this mess doesn't make it better. So I was trying to think of what story am I trying to tell. I have important information to get across. Bacteria levels are different in wet and dry weather. Where is each site located relative to the other, relative to the person looking at the chart? Provide some colloquial information about sites. Don't use a string of letters and numbers, and use a common name. I saw that in the charts earlier too, so happy to see that. I think that's one of my pet peeves is seeing charts and it's just a bunch of letters and numbers, and you have to look at a different chart and find it. And I learned a couple of years in that even my volunteers didn't necessarily know what their site IDs were. They just copied it off of their instructions each week and didn't understand what the numbers meant. So what I thought was really helpful information maybe wasn't. And then also, what are the standards? When and where are they being met? And what does that mean? So the tools that I have found the most helpful is clear, familiar color coding systems, having your thresholds clearly marked on the graph, explaining what they mean. And in this case, I also chose to compare a simplified map with the graph. And then the bonus, use some tech skills to generate an interactive graph. And so here's what I came up with. And so on the left, we have the Connecticut River, which people who are familiar with the Connecticut River, they might recognize that, and that would actually be helpful because some people might know, oh yeah, I live, in this bend, or here's Massachusetts, here's Connecticut and Vermont. And then I use stoplight colors, thankfully no reds. And then these are also colorblind friendly, that's why they're a little saturated. So that's if someone was colorblind, they could see the difference between the colors. Clear demarcation of the standards, and then CRC tends to, instead of saying primary and secondary contact recreation, say swimming and boating, which you guys also did as well. I use short common names for each site. And then even when I don't have a map, so these are ordered from upstream to downstream. Even when I don't have a map, I tend to order sites by upstream to downstream because it helps people see the profile of the map. And this is, so it is interactive, so if you put your mouse over the chart, it highlights it on the river and vice versa. Because this was published online. And I'll send a link to this presentation and you can click and see the rest of the report. But also when I'm presenting, I found that I often have to present data in 2 or 3 different ways to tell the whole picture, because looking at averages might tell one story, and looking at box and whisker plots might tell another, but they're kind of confusing. They're really useful, but still confusing. And, and looking at geographic profiles versus percent exceedances and things like that. So presenting data in multiple ways can also be pretty helpful. All right, so that was one way of communicating. And then also, what I was actually asked to talk about was revamping our the Connecticut River Conservancy's Is It Clean website, which I was involved in from the very beginning. So the website launched in 2012, which if you remember, that was the year I was volunteering in the lab at CRC. And so I was there at the very beginning. They were finishing developing. I was like testing bugs and having to enter things in multiple times. It was originally built as a partnership with the Pioneer Valley Planning Commission, which is the planning commission for the Hampden and Hampshire counties. And the goal was to showcase the E. coli data being collected throughout the Connecticut River watershed, in addition to CRC and PVPC at the time, NepRWA, the Millers River Watershed Council, the Farmington River Watershed Association were early adopters, quickly followed by Ashwillet, the Chicopee Four Rivers Watershed Council when they started, Scantic River, and Black River Action Team. So it was well adopted. It was always planned to be bilingual. So everything was supposed to be available in English and Spanish, and had a side mission of serving as CRC's lab's water quality database. So the thinking was, we're building a database for this website. It will also be the home of the data. And so this was 2012. It was somewhat high-tech at its development, but it had a lot of custom code because there just wasn't the same geographic data tools that we have now. And it was built in Drupal, if you're a web nerd and want to know about that. right, so I'll show you a couple iterations of it. I had to use the Wayback Machine because I didn't have screenshots of the old website, but I was able to find most of what I wanted to show you. And so this was the original website, and you know, things are a little wonky, like the map is in 2 different colors, but we're lucky it works at all. And so it was incredibly helpful. It was also not very mobile friendly. Now I've broken the map. And everything was in alphabetical order. You couldn't really search it. We had some issues where like someone spelled out Vermont and also used the abbreviation. So there were a few challenges for it. Can't believe I broke the map, which had the pins, but the pins were color-coded with that. And then, as you can see, there was other stuff on this website about recreation in the Connecticut River Valley. And then this is what a site page looked like. We had, you know, a picture. Sometimes this should be a map. We could link it up to a USGS gauge site. One of the initial confusing things is, you know, in our database we just said wet or dry, which is had it rained recently. But they didn't write wet weather, they just wrote wet. And so it turns out some of our early partners were marking all samples as wet because they're water samples. But yeah, so this was what the site was. And, you know, some positives, it had a static URL, so we could set up QR codes at sampling sites or share specific sites. You know, people could bookmark their favorite sites. 2022, the map. This map doesn't want to work at all, but you know what Google Map looks like. We were able to take over the site and rebrand it to match DRC's branding, made a few tweaks, but we still had some of the same challenges, which was, you know, all of our sites were just in alphabetical order, and we had a lot of sites because we have the whole watershed. We still had some of the like duplicate state issues. And then we were trying to show like that some data was old because we had a few years where like it rained on the last day of sampling and then the pins were red for until the next year. And then this is what a site page looked like. You can pretend there's a picture. The data isn't connected, but instead of saying wet, it did say wet weather. And then there was a little bit of text that was generated using some statistics about how often the site tested exceeding the thresholds for swimming and boating, but it was never really explained. You might have noticed the latitude and longitude swapped. So we definitely had some challenges. So one of the big challenges that we had, and I think that is a challenge for most web development, is that funding comes to develop a fancy new website. What? Sorry. Oh, I don't think you could hear that, but I started playing a video. So funding for ongoing maintenance and update just didn't really exist after the initial development phase. We had a hard time communicating with our web developers about water quality. For example, the lat and long were switched, and we couldn't figure out how to like convince them. Like, it was never a problem that they saw it. So even though we asked for it to get fixed, it never really did. And then we switched web developers, and they didn't understand the custom coding behind the website and couldn't make any changes. So that's what we were dealing with. And so decided the best approach was to start from the ground up, rebuild the whole thing. So we worked with the Commons, which is a nonprofit web group, nonprofit environmental technology firm, and they helped us identify the tools that would work to meet our goals. We moved all of the CRC water quality data out of 500 Excel files into Airtable, which is kind of like a fancier Google Sheets with some pretty powerful database technology and tools. And one of the big things is we separated the is that clean partner data out of our own data, because that was another problem with using a website as a database when other people could contribute to it, is it wasn't actually serving that purpose. And with the Commons, developed a new vision for the website, created an RFP, and then we worked with Fedo Studio to develop the website itself. They had weekly meetings from wireframes through, we ended up doing more custom code development. But thankfully, one thing that they did is their philosophy is that they'll build the initial website. They want to provide all the tools so that they can walk away when you turn it on and the organization running the site would know how to do anything. They could hire any web developer in the future who could read the documentation and make updates. And they really took the time to understand water quality data and what we wanted to communicate. I think that made a huge difference in the process. So, the new website is much shinier. It's at isitclean.us. It has location, so it shows, you can see I'm 3.1 miles up from the mouth of Mill Brook. So you might know where I am now. And we're able to show, one of the big things that we talked about was that people want to know what is the most likely condition and not what was the most recent condition, because some sites are sampled every week, some are sampled every other week, some are sampled monthly, and then some are, and then we don't sample for the entire winter. And so, after lots of conversations, one of the things we decided to do was show just a basic statistical calculation based on the percent of times that it's been tested and exceeded a certain standard, and the most recent weather. And so we can look at the latest samples. These were collected last October most of the time. The little ones are really old, and so we're able to address the too many gray and black pins by creating still color-coded pins, but different sizes and emphasis on the map. But it mostly hasn't rained recently, and so we see a lot of blue. If there's a big rainstorm, so maybe come back tomorrow or Thursday, and I bet the pins will be a much different color. And one of the things, you can click on a pin and see about it. So it hasn't rained. You really should be swimming right now. It's very cold and icy. But if it was a nice summer day, 89% of the time on a nice dry day, the Connecticut River for Chulski fishermen access meets the standard for swimming. And so I'll click on that. And we still have the static pages. We thought that was really important, being able to have a QR code or have a volunteer be able to bookmark it. We were able to assign site uses. One of the things is this has public access. We test a lot of private property, and we still think that information is important, but this is much clearer to communicate that it's private property and it's for informational purposes only, but this is a nice boat access, so people fish, people boat. We have the, it's not real-time data, but based on all of the 84 measurements that this is taking into consideration 75 times. And if there is a really clean site, it will still only say 99 chance. Because we can't be 100% sure about everything. Very clear icons to communicate dry and wet weather, the colors, and then, for the people who really want to know everything, they can go and keep going and keep going. Map. Who's sampling? What other sites are nearby? And then if you wanted to see so tomorrow we might come back after we get a bunch of rain, and we'll see that there's only a 62% chance that it's clean for swimming. And so that might be, that allows the user to make their own decision. And one of the big things that I loved about this project is that it was a real partnership. Lots of groups, government agencies like USGS for a while was sending us their data, their E. coli data to update, and just so many partners, contributing so that we have this huge map. That's a lot of sites, that's a lot of geography. There's still plenty of a watershed up here that could get covered, but it was definitely a great project. It came out better than I could have imagined, and I'm happy that it was one of the last projects that I was able to work on at CRC. And so I'm actually going to stop my screen share so I can see all of you, but happy to answer questions.
Are there any questions for Ryan? I'll try and convey them as best I can. Hard to see. Warren?
Did Ryan do all the work himself, or did he have a team of people to do this? How long did it take, and what was the cost in man hours, and any contracts that helped to develop that?
Yeah. So, Ryan, I don't know if you could hear that.
Yeah, I could.
Okay.
So no, I did not do it all myself. It would have taken me much longer, and it would not look as good. Like I said, we worked with the Commons, which is the nonprofit, environmental science web development firm. They are behind. They used to run Water Reporter, and they have some stuff called Field Docs, and now they focus on working with mostly nonprofit groups to create the tools that support their work, and then hired so we worked with them for about 6 to 8 months, I can't quite remember, to identify what our goals were and pick out the tools, and then they did help us migrate all the water quality data into Airtable and set up different workflows and formulas and stuff to make that all speak to each other and in the future speak to a website. And then the actual web developer, because we were doing something kind of really ambitious, so we hired an actual web developer, Fedo Studio, that did a lot of custom coding to put that map together, to scrape weather data so that the map knows whether or not it's rained recently. And so with them, we met weekly, sometimes twice a week, reviewing how things are working, all of that. So then, I don't know how many hours it took all together. I think the contract with Fedo Studio was 400 hours. I don't remember with the comments. It was quite pricey. We were very fortunate to have a private funder who prefers to remain anonymous. And he also had some experience with websites, having funded them, and so he also sat in on a lot of the meetings to help make sure that they avoided pitfalls that he had encountered in past projects that he had funded. And So it took a lot of hours, and it took, I think, $80,000 or $90,000 total. But it's we hit a lot of the like pie in the sky goals, like that real-time map that takes into account whether or not it's raining recently. Because I think that is one of the things that has been the hardest to communicate over my career, is that it's not the most recent data point that's important, it's the trend of what happens based on the weather, if you're trying to decide whether or not to swim.
So given the significant investment you put to developing that, and that some of it is kind of a nice new, the weather scraping and the map representations, is any of that, did you guys open source that, or is that something like, is that framework something you would share to other watershed associations? Do you have plans to sort of expand on that, or be able to take that to other audiences as a framework?
I think that while we were working on it, that was definitely something we were thinking about. I will send out, or I'll send Sean my presentation so he can send that out a follow-up email, and there's some links in there. And the Commons did a case study explaining exactly the whole development process, and I think it's something they'd definitely be interested in replicating. And or expanding. So yeah, it was kind of like we just developed the coolest website ever. I wish everyone could use it.
Anyone on Zoom want to ask questions?
Excuse me.
Yeah.
So, again,
Yeah.
Somewhere you ask this question, what results are good, what results are bad? What do you mean by this question?
What do I think about that question?
Yes.
I mean, I think that's what people want to know. I think the truth is that the results are nuanced and they tell a lot of information. But I think that most people want to know, is the water good or is it bad? And it's not always an easy answer, but in a case of something like E. coli, the EPA and the National Health Department and people did a lot of research to figure out the numbers that hopefully inform safer recreation. And so that is often what I'm trying to convey. Is it meeting a standard? Sometimes the standard is lax, and so you might have found another value in the literature to give it a little, be like, well, it's meeting a standard, but some studies show that we should be aiming for this concentration or something like that. So with E. coli, it's a little easier to say this is good, this is bad. Don't get it in your mouth, don't go near it, are the two levels basically. And but I think that's the question that a lot of people have. And so I tried to figure out how to answer that question, and not just my own questions.
But I think at the level of a river, so this result, for example, the presence of E. coli is for a short time, because that can change, anytime during the day. So, due to the rules of the Environmental Protection Agency, is the protection of the water ecosystem. In this case, it's very important that we take measures to protect the ecosystem.
Yeah, I think that's a good point, and I think one of the best things about the website they've developed, to speak for Ryan a little bit, is the fact that it's showing not just what the last result was, but what you might expect if you went there today, even if there's no sample taken, based on previous results. So if it hasn't rained in a while, you can look at the past dry weather samples and say it's a good chance that it's safe to swim, even if they didn't collect a sample today, right? So
Even at the website, is the problem of water. Okay, the presence of E. coli is not permanent.
yeah,
It's for a short time. What we have to do is protect according to environment. What we have to do is, in the case of the climate change, what we have to do to protect the water.
yeah, that's a good point.
I think on that point, I think that's one of the balances too, because I think a lot of people have very short-term interests. Can I go swimming tomorrow, or this summer? And a lot of the more boring questions are, what is climate change doing to our rivers? That's a much slower process of study. It's much more math intensive. So yeah, that's a challenge, meeting all the different questions that people have about water and how it affects them, how it might affect their kids, how it's affecting the wildlife. There's so many. It's a complicated topic for sure.
Sean, I have a question. So the data, the percentages that you see when you click on a site is the average over time. How often are those updated with new data?
It depends on the site. So CRC's main STEM sites are sampled every week from the end of May through the beginning of September, and then some organizations sample every other week. There's a few that sample monthly, but the way that the Airtable is set up, I think at 8 p.m. each night, it checks for new data and updates it.
Oh, so it changes literally day by day, week by week then?
Yeah.
Oh, wow.
And it's constantly checking for rain.
Right. No, I got the rain part, I was just thinking.
Yeah, so the actual answer is, as soon as somebody uploads it.
That's cool.
All right. I know we're a little over time. Thank you all for coming. Thank you both to Meher and Ryan for great presentations. If you guys have more questions, we'll be reaching out with the slides and the handouts, and we'll definitely be in touch, especially all you volunteers. We're going to round you up in May, or round you up in April, so you're ready for May. And thank you all for coming.
Thank you.